Good morning, Ronald. Shanili, Akshaya, Mridula, Vishalini, Arshia. Very good morning. Okay. So, when we see this lesson, mineral nutrition in botany, that in that mineral nutrition you will find most of memory-based content only. There were very few, maybe 5% conceptual basis are there. Remaining or memory based. We'll try to deal that lesson little bit later, maybe tomorrow. Okay, so first we will deal with two important chapters. Good morning. So first we will deal with the two important chapters, right? Those are respiration in plants, and then we'll see what are the important aspects you need to cover in this respiration in plants, and we'll see the other lesson, right? Excretory products and their elimination. Okay, so these are the two things, two chapters we'll cover today. Okay, good morning, Abhiniti. So let's see that what are the important aspects you need to cover within these two lessons. Okay, so first we'll start with respiration in plants. So in this respiration in plants, most aspect you need to focus on reactions like anaerobic and aerobic reactions, like cellular respiration we'll talk about here. So breathing is concerned, right? The cellular respiration we'll be talking, but the exchange at the level of stomata, the gaseous exchange of this at the level of stomata is little important. But when compared to this cellular respiration, that we can divide into aerobic and anaerobic, and those equations are important. Okay, so what are anaerobic respiration products? End products of anaerobic respiration. You can get lactic acid or ethyl alcohol, right? So there is the organic food material is not completely burnt or not completely oxidized. So this is something catabolic reaction. So we have got formation, photosynthesis. We already talked how glucose is formed, and here we will see how glucose is utilized. So mostly we will be talking here about mitochondria, cristae, and their ATP synthase. This proton pumps, right? Especially this ATP synthase, which actually creates the ATPs, right? So which helps in the synthesis of ATP. So that we will try to talk. So especially when we talk about aerobic and anaerobic, sorry. So when we talk about aerobic and anaerobic, okay, so we will be mostly focusing on their energy consumptions. So how much energy will be utilized during aerobic respiration and how much energy will be utilized during anaerobic respiration. Okay, and how much energy does released also we need to focus. So when compared to energy release in this aerobic and anaerobic respiration, you will find in aerobic respiration you will find more of an energy, right? Almost 680 kilocalories per mole would be released in aerobic respiration compared to anaerobic. Anaerobic only 21 kilocalories or more or less 20-25 kilocalories of energy will be released, because here the glucose molecule is not completely oxidized, because it is converted into some other products like ethyl alcohol or lactic acid. Lactic acid in the case of muscles, right? If I keep my hand like this for a while, right, what happens? My hand would pain. Why is that? Because the muscle is actually releasing this lactic acid. Anaerobic respiration takes place within the muscle cells, and then finally it leads to release of lactic acid, and lactic acid actually brings up the signaling of fatigue. So these are the things: how much energy utilized, how much energy is released during this aerobic and anaerobic respiration. that might be the potential questions. Okay, and you need to focus on one thing is that what are the products or what are the substrates of what are the substrates of this respiration. So if you see the respiratory substrates, we have carbohydrates as carbohydrate and fats or as an respiratory substrates. So which molecules are actually used as an substrates? Substrates, enzymes gives rise to. So that is the equation, right? Every time we need to write. So here we are actually talking about so this is enzyme plus substrate, enzyme substrate complex, enzyme product complex gives rise to enzyme plus products. Okay. So here we are talking about this. So here what are the substrates? Respiratory substrates. So respiratory substrates vary. So respiratory substrate can be a carbohydrate or fat when it comes to floating type of respiration. And protoplasmic respiration, if you see, proteins are used as a respiratory substrate. But here we are not going NCERT. They didn't cover these types of respiration, but for your information. Okay, and the next one is that you need to focus on the respiratory coefficients. So respiratory coefficient, how to find out the respiratory coefficient that you need to work on. So if you see the respiratory coefficient RQ, how do you calculate this RQ, respiratory coefficient? For that matter, what is the respiratory coefficient? You might have come across this, right? So what is the respiratory coefficient? Can you recollect respiratory coefficient? So this numericals related to respiratory coefficient might be, expected. Okay, respiratory coefficient equal to volume of CO2 walled by volume of O2. So here CO2 actually, CO2, O2. Okay, not COO. So this formula you just try to memorize, and you see that what exactly the respiratory coefficient is a ratio of volume of CO2 released to the volume of oxygen absorbed, right? So this is the respiratory coefficient. You may expect this type of numericals in this chapter. And respiratory coefficient varies, when it comes to different plants. Okay, so succulents is different, and anaerobic respiration it is different, right? So that you need to focus on. So coming to the aerobic respiration and their pathways. So we have in this lesson 3 important pathways. One is glycolysis, Krebs cycle, and electron transport chain. So here in the glycolysis, like the glucose is actually lysed, dissolution or breaking down, right? And it is also called as EMP pathway. So EMP is nothing but the scientist names. Once you try to recollect the scientist names also. Okay. So the EMP pathway, or you can see, just I'm trying to type this. So the scientist names I'll try to remember. So glycolysis, where does this glycolysis occurs? The the position, not position, actually the location of this glycolysis. It occurs in cytoplasm, cell cytoplasm, and this is the major pathway where ATP synthesis would be present, right? Especially if you see this glycolysis occurs in mostly the tissues which are lacking mitochondria, like erythrocytes, right? Erythrocytes, cornea, lenses, etc. This glycolysis occurs, and finally we will be getting ATP. So you need to know that where exactly glycolysis also occurs, in which type of cells, right? So the reactions of this, glycolysis, the entire balanced equation, and you have to focus on what are the, enzymes that are involved in this, EMP pathway. Okay, what is the first enzyme involved in the conversion of glucose to glucose 6 phosphate? Can you guys recollect? Can you guys tell? What is the first and foremost important enzyme? Okay, that's nothing but hexokinase, right? I told that this is not completely full length class. Otherwise, I would have been explaining each and every aspect. But as already these lessons over for you guys, you please try to recollect. Okay. So if you see here, glucose is converted to glucose 6 phosphate, right? You know what is 6 here? 6 phosphate IUPAC. You can really understand what 6 signifies here. So exokinase activity would take place. Like that, each and every step there is an interconversions of this glucose molecule, and finally this 6 membered ring, this 6 carbon molecule would eventually split into glyceroldehyde 3 phosphate, 2 molecules, right? So this glyceroldehyde 3 phosphate is further transformed into pyruvate. So this pyruvate of 2 molecules are the end products of this glycolysis, right? So after secreting this pyruvate, you need to find the net results of the glycolysis, right? So what are the net results of the glycolysis? The end products of like 2 molecules of pyruvic acid, where this pyruvic acid goes into the next cycle, right? It involves in the next cycles. Nothing but the Krebs or tricarboxylic acid cycle, or Krebs TCA cycle, where the pyruvic acid is the initiator, where it starts the entire chain of the reaction. So 2 pyruvic acids would be the end products, 2 molecules of ATP and 2 molecules of NADH2 and 2 molecules of H2O. So total 4 ATP will be released from the ATP itself, from in the form of ATP, 4 ATP, and from NADH2, Na1 NADH2 equal 3 molecules of ATP, so it is 6 ATP. So total release is 10 ATP molecules, but how many ATP is consumed is 2 molecules. So the net gain or net yield of glycolysis for one glucose molecule is 8 ATP, 8 ATPs. So this is how we need to calculate the energy, right? So these aspects are important when it comes to this glycolysis, and the enzymes and their functions are utmost important. And the metals that are metal ions, or the ions which are present in this each conversion. For example, glucose and from glucose to glucose 6 phosphate, right? There is Mg2 plus ion is involved, right? Where exokinase activity would be taking place, and you will find that glucose 6 phosphate would be forming. And from this glucose 6 phosphate to fructose 6 phosphate, there is an isomerization. So this isomerization takes place by isomerase enzyme. So phosphoexose isomerase. So this is the enzyme. So this is the enzymology nomenclature is very easy when we can really relate to the what kind of a reaction is taking place. Okay, so you need to focus on in this EMP pathway what are reversible and what are irreversible reactions. Maybe this would be the potential question. So there are some irreversible reactions, right? And there are some reversible reactions. So reversible reaction, for example, aldol condensation. So aldol condensation would be taking place here, where the two 3GP molecules are formed here. So that would be the reversible reaction, right? So you need to focus on which are irreversible and which are reversible reaction. Yeah. And what else? In this, you need to more focus on in which stages ATP is generated and NADH2 is generated. So these are payoff where eventually leads to formation of ATP. So you need to focus on payoffs where exactly we got these energy molecules that you need to concentrate. Okay, I'm just giving the highlights where you have to focus on, right? So where exactly the molecules are actually coming, right? Molecules are actually especially these energy molecules. So what we are trying to understand is how the energy is produced, how the cell is respiring in the case of plant. So or even in the case of muscles, etc. So how much energy is producing, where in which reaction this energy is producing, payoff reactions, reversible, irreversible, and the end products of glycolysis is important aspect. And then coming to the next cycle, that is Krebs cycle, right, or tricarboxylic acid cycle, right? The entire equation you need to focus, you need to practice this entire equation so that it would be useful. So what are the substrates involved in it? So what are the substrates involved in it, right? What are the different enzymes involved in it, and whether it is aerobic or anaerobic pathway also you need to focus. So when you see this Krebs cycle, it acts as an amphibolic pathway. What do you mean by amphibolic pathway, by the way? So amphibolic pathway is both, it can act as catabolic or anabolic, right? So if you see this Krebs cycle, it is a series of 8 reactions which occurs in aerobic environment. So the cycle which is given, where first pyruvic acid is actually how it is reacting with the this acetyl CoA to form oxaloacetate. So these are the first reactions and citrate formation. And in this entire cycle, you guys have to focus on where how many molecules are utilized, how many ADP molecules are utilized, how many produced. So what is the net yield that we will be? You need to focus on it. Okay. So this Krebs cycle can be inhibited by 3 different molecules. So this is not, I think this was not covered in your NCERT. So I'll be giving here itself. So what are the 3 molecules, 3 inhibitors of PCA? If you see those, fluoroacetate, arsenic dehydrogenase, and malonate. So these are the 3 important inhibitors which can inhibit this TCA, so that this TCA cannot really happen. So once this TCA is not happening, the products of this TCA would not form, and finally the normal functions of the cell or the plant would not be taking place. So might be they may ask that what are the 3 different or what are the inhibitors that you notice in this TCA. Okay, so I'm just giving here. Okay, so try to take notes, don't forget about this. Okay. So enzymes that are involved in this TCA, citrate synthase, right, isocitrate dehydrogenase, and their functions. Alpha ketoglutarate dehydrogenase. It means what exactly they are doing, whether they are actually reduction is happening or oxidation is happening, how many ATP molecules are forming, where hydrolysis is happening, or formation of H2O, release of H2O2 is there in which steps. And if you see FADH2 formation, right, this FADH2 formation also you need to focus. So especially the output of Krebs cycle is almost for 1 molecule of pyruvic acid through Krebs cycle, it yields an equivalent of 15 ATP molecules should be produced. So that's what the importance of this Krebs cycle, right? So the glucose is not directly utilized or directly not producing the entire energy, but it is first given as an end product pyruvic acid, And to form pyruvic acid, 2 molecules of ATP is utilized and 8 molecules of ATP is released. Means yield, net yield. But that's not sufficient, and still this pyruvic acid undergoes further, oxidation, and finally it leads to 15 ATP molecules, right? And then comes to the ETC, electron transport chain. You need to focus on these complexes, different complexes that are there. Complex 1, 2, 3, 4, 5, 5 complexes and their functions, right? So these 5 complexes you need to, at most you need to focus on these 5 complexes, right? So I'm giving the 5 complexes also. I'm just giving a recap. So in electron transport chain, these 5 complexes might be given as a section A and section B type of question. So complex 1, the corresponding enzymes or corresponding functions. So you shouldn't etc. You shouldn't be ignoring at all. Okay. So if you see this 5 complexes. These are the different, these are the 5 complexes you see in EPC and their functions, right, are at most important. So at the end of the aerobic respiration, what are the end products that we get? What is the yield, net yield we get? So how many molecules are actually we got from each of the cycle? For example, if you see the summary of the aerobic respiration, that you need to focus. I'll be sending that in your WhatsApp group also. You try to see that. Okay. So if you see this, what is a summary of this aerobic respiration is also important. So I'll be sending that in the group. Try to look into that. Okay. So right now I am sending it. Okay. So this summary is important when it comes to the entire three type of reactions. So I just sent in your WhatsApp. Just try to have a look at it, right? So glycolysis, how many ATPs are utilized? How many molecules are produced, right? How many pyruvate? This pyruvate is oxidized to yield how many NADH2 molecules? NADH2 molecules, and the Krebs cycle formed how many NADH molecules, how many protons, how many FADH, everything. The summary I've already sent. So this is the focus where you need to keep. So don't ignore. Okay. And then you have fermentation, and PPP pathway also is there. Pentose phosphate pathway. So if you consider all these reactions, you need to focus on how many molecules actually utilized and how many molecules that are not being utilized. So that is the focus you need to have. Clear? So any pathway you can concentrate, right? This is the framework you need to follow. So how many molecules are utilized, how many molecules are net yield, this is the thing you need to focus. Either fermentation or glycolysis, anything you take. And when we talk about the fermentation products, okay, 1 minute. Bike you take. Yeah, so sorry. So that is a thing you need to focus, especially when it comes to fermentation. There are different types of fermentation, and their fermentation products also you need to focus. Is that clear? So these are the aspects that you need to focus in the respiration. Okay. Hope you guys are following. What are the important aspects that you need to focus? I'm just trying to enumerate in this, right? So these are the things which are covered in your NCERT also. So when you see these, different, all these reactions, you need to more focus on the enzymes and the substrates. First enzyme, first substrate. What are the how the cycle started? Right, so those substrates are involved. For example, for glycolysis, glucose is substrate. For TCA, pyruvate is the first initial molecule, and then oxaloacetate, and then citrate formation, etc. So like this, the first initial substrates and enzymes and cofactors, right, or other ions. So that's what this metabolic reactions are not that easy, right? You need a lot of explanation to this also. I hope you got it in last year, but if not, what I can do is, if you have any requests, I can actually do a separate videos. Right now, it is not possible within a 1 hour. No one can actually cover these reactions and all, right? So if you have any request, you can ask me so that I will make a video on a complete lesson as well. Okay, fine. Otherwise, if it is not needed, you can just go through the content. So these are the highlights that you need to focus in this lesson. Okay, and coming to the next aspect, next lesson, that is excretory products. Excretory products and their elimination. When you see this excretory products and elimination, so obviously the examples of these excretory products, like or animal types, amniotic, uricotelic and ureotelic, and their examples and their products. So if you see this aminotelic, where ammonia is released as an excretory product, ureotelic, urea, and uricotelic, uric acid. So which animals does you see this type of excretory products? You need to focus, right? And you see this excretory products of different animals across the kingdom Animalia, you should be focusing. And coming to the excretory system, you have got structure of the kidney, and most probably, the nephron diagram would be the more important than the kidney diagram. So the nephron and its in it and its parts. So it starts with a Bowman's capsule like this. So Bowman's capsule will have capillaries, right? So glomerular apparatus will be, the glomerulus will be present. And what are those functions? So right from the proximal convoluted tubule, distal convoluted tubule, and then there is a ascending and descending loops. And this structure is utmost important because you may get confused with the positions or the parts of this, where they may be asking descending limb would have whether reabsorption or absorption phenomena would be seen in descending limb or distal convoluted tubule. That you need to narrow down. That's it. So these functions and these, especially this parts of the nephron is more important. And when we talk about the nephron and whatever is present in the NCERT, especially the tables and the diagrams, you should not be neglecting. So when we talk about nephrons, right, these types of nephrons, there are cortical nephrons and juxtamedullary nephron, where they present and what is the percentages of these nephrons and what are those functions. So what is the main difference between cortical and juxtamedullary nephrons also you need to focus, right? So urine formation would be the more important, and during urine formation, right, calculation of GFR, glomerular filtration rate. So GFR, how do we calculate? So filtration, this GFR calculations, or what exactly the GFR, right, present in a normal healthy person, right? For example, that is 125 ml per minute is a GFR, right, or 180 liters per day. So what is a GFR actually? It is a quantity of glomerular filtrate formed per minute. So we know what is glomerular filtrate. So once this blood is actually going through this nephrons, this glomerulus would actually filter that much of volume of the blood. So that is what we call glomerular filtrate, and when we add the time for it, the change in time in perspective, we can see that glomerular filtrate rate. Clear? So there is a glomerular filtration, selective reabsorption, and tubular secretion. So these are the three important aspects in urine formation, and each aspect is utmost important. Where in ultrafiltration taking place in which part, and reabsorption is taking place. In reabsorption, which ions are reabsorbed, right? And secretions, which are secreted. What are the different hormones that are responsible for this? Secretions also you need to focus. Okay. So the concentration gradation, that is osmoregulation, takes place, and especially, for example, when the volume of blood is increased. So what happens? What happens to the filtration rate? At the same time, how that body fluids volume is actually taken care by this excretory system. So when the volume increases, how this system would be excreting. So how they are actually releasing, and when the volume of fluids in the body is less, and how they are reabsorbing from the urine to make it more concentrated. In which parts of the neuron that reabsorption takes place, that you need to focus. Henle's loop is important, and counter current mechanism is important. So if you see this, the urine, this urine formation within this, for example, within this nephron would be in one direction, and the blood flow in the capillaries associated with this neuron would be in opposite direction. That makes it counter current mechanism. Okay, so especially the vasa recta and Henle's loop. So this is where counter current mechanism would take place. And what is the importance of this counter current mechanism? Also important, and more importantly, what you need to focus in this lesson is regulation of kidney functions. So this regulation is key aspect of this entire lesson. So this is a hot topic in this entire lesson. That is, how do we first we talked about anatomical aspects, where exactly these nephrons are present, what are the different types of nephrons, and how do these nephrons actually form urine, right? That urine formation and different hormones involved in it, right? So juxta medullary nephrons and cortical nephrons, you see that hormones will be released, and how these hormones will be regulating the this GFR also you need to focus. So this regulation of kidney functions is utmost important because the hormones do involve, right? So controlled by juxtaglomerular apparatus, JGA, juxtaglomerular apparatus. How this would actually really increase or especially when we talk about renin-angiotensin mechanism, right? So how this JG cells actually release this enzyme renin, and once this renin is actually going into the bloodstream, how they are acting on the blood vessels. So this is renin-angiotensin mechanism is utmost important, or else we can call this as renin-angiotensin aldosterone system or mechanism. So this is very important. So I'm giving what are the important aspects that you need to focus in. Renin-angiotensin mechanism, how these blood vessels are constricting and relaxing. ANF, right? Atrial natriuretic factor, and how this ANF would actually released by the atrial walls in response to the increased blood pressure. What exactly they do? So how they are interacting with the JGA, that you need to focus. At the same time, antidiuretic hormone ADH. These things we will be talking in the chemical coordination also, but you need to focus in the functions of regulation of this kidney functions. Okay. And what are the other organs that does this involved in this excretion also? You need to focus. And finally, disorders of excretory system, glomerular nephritis, kidney stone formation in kidneys, right? And renal tubule acidosis. So few few of the excretory system disorders were given in NCERT, but if you go through your NEET book, you will find n number of disorders. So you kindly just try to cover those disorders as well. So what exactly the name of disorder? What exactly the happening over there? For example, kidney stone. Stone is formed, right? Where what is the location of stone formation? It can form in renal pelvis or it can form in ureters, right? So what are the symptoms, right? These are the things you need to focus on, right? So when we talk about this excretory products and excretion in human, right? These are the important aspects you need to cover. Okay. So these are the two lessons when we talk about the two lessons, and let's see the one more lesson, that is mineral nutrition. What are the important aspects you need to focus on the mineral nutrition? In mineral nutrition, I told it's more of a chapter 12. When we talk about chapter 12, is more of a memory based question should be coming. So you can expect very less number of questions from this. That's why I kept you know last. So you can, I have already forwarded 30 days of NEET preparation ebook, right? PDF, right? So you can see the even weightage of each lesson topic wise also. So you will have a more glance. Anyhow, you will be having a lot of time after grand tests also. So you can revise a number of times. Okay. So that is one of the important aspects that we went on YouTube Live, so that whenever you have doubts, you can go back and see any number of times. So when we talk about this chapter 12, we have classification of mineral nutrients, right? So we'll try to stop here. I think I took more than I actually went off the schedule. Okay, let's stop here. I'm thinking it's a 12 o'clock. Okay. I think it's 9:50 to 10:50, right? So we have time actually. I was a little confused of me. Okay. Yeah. So 11 o'clock is your physics class, right? So we have a little more time. So 20 minutes time, we'll see that this 12th chapter, what we can cover. So in this mineral nutrition, you need to focus on what are essential and non-essential mineral elements, micronutrients and macronutrients, their examples and their functions, right? And coming to the deficiency of this mineral nutrients, what happens when these mineral nutrients deficient, essential mineral nutrients, right? Chlorosis would come, necrosis would come, wilting, etc. You can rotting, or you can see this when these mineral nutrients are actually deficient. But when they are increased in their concentration, it may lead to even toxic concentrations. So what are those examples, right? And in this, you have got metabolisms of different nutrients. When we talk about nutrient cycles, we have nitrogen cycle here. So nitrogen cycle, you need to focus on what are the different usable forms of this nitrogen, right? Nitrate, nitrate, ammonia, etc., and how they can be utilized and how they are produced. What are the different reactions, or what are the different organisms that's involved in this cycles that you need to focus. When we talk about nitrogen and symbiotic, you have got rhizobium and lichens, so and how they actually fix this nitrogen, you need to focus. So what are the different uses of this nitrogen, right? At the end of the nitrogen cycle, what are the different products that we observe, also you need to focus. So these are mostly of memory based concepts that you can really go through by yourselves. When we these experiments, or the methods or experiments that are given, also you need to focus on them. For example, you have one of the method, hydroponic culture. So what is it, and what is the aim of this hydroponic culture? So why we are using these systems, right? And how can we really how can we say that this uh, nutrients present in this plants would really matter, right? So what are the different types of experiments you can really do to understand that nutrients are essential for the plants? As simple as that. And role of micronutrients and macronutrients in the physiological functions also you need to focus. Deficiency and toxicity levels, and how do they absorb in transportation plants. Also we already discussed how the absorption takes place, right? And this you know when comes to this cycles, we have given only one cycle, that is this nitrogen cycle. How this nitrogen cycle is utmost important, and how this nodule formation, root nodule formation takes place, and how that is helpful in the case of nitrogen fixing also, that you need to focus. So these are the main important aspects, nothing more than this. So what do you need to focus is what are micronutrients and macronutrients, and their examples and their roles, and their if they are deficient, if they are high. So these are the aspects you need to cover, especially nitrogen cycle, nitrogen fixation. You will be coming in even in ecosystem chapter. You will be talking in even human health and microbes in human welfare. So more or less, you organisms and population. When we talk about these fertilizers, right? These will keep coming. So what are the things you need to focus is micro, macro. There, what are the substances involved in it, right? Molecules, compounds, and their functions, roles, deficient, and high. So these are the important aspects you need to focus in this mineral nutrition. More than this, nothing conceptual, but mostly you should be focusing on the tables that were given in the NCERT compositions, right? I'll try to do a video on the mnemonics on this, remembering how to real memory techniques of remembering these compounds, right? So what is the composition of carbon, nitrogen, oxygen, phosphorus within the body systems, living systems? I'll try to do. So these tables, don't ignore the numbers, or you need to practice, memorize at any cost, right? So this is the chapter where you can use your memory, but you need to have some memory techniques so that you will not have stress to memorize them. Okay, but understanding goes little in this. It means how much ever you understand, if you really don't have a memory technique, you can't really remember this compositions. Right, except this lesson, other lessons you need to understand, and you cannot really go by just memorizing things. Okay, with this we'll end this session, where these are the 3 lessons you need to focus, right? The mineral nutrition, we'll try to see what are the highlights we can cover in the next coming lessons. And I've sent a photograph, that is what are the respiratory end products, right? That is given in the WhatsApp, our class group also. Try to focus on this. So let's end this session. We'll see you in the next class, right? And if you have any request to you can send me your request to take any topic, so that I will be explaining that topic. Okay, I can't keep explaining the entire chapter right now. So if you have any request to explain, sir, can you explain this topic? I will make a video on this, and then I will post maybe a 30 minutes or 20 minutes, 25 minutes. Topic wise, I will try to make it. But it depends upon your request and your interest, at the same time, your requirement. That's it. Okay. Thank you. See you in the next class. Stay home, stay safe. Thank you.